Dear judges, instructors and students, good morning. I'm Brittany He, today's host. Our project is to test environmental hormones. So, what are environmental hormones? Exogenous endoserine disruptors are called environmental hormones. EDCs are compounds that affect metabolism. More than 30 types of endoserine hormones were detected from the sipping water at the industrial waste disposal site. Therefore, in order to prevent more babies and children from being harmed by precocious puberty, we choose to study this topic as our hygiene research project. At first, since we have limited access to information about environmental hormones, our team member decided to consult professional professors for more information. The follow is a report from Tom Tsai.
Well, thanks to Brittany, and now I will introduce the interview. Due to the lack of knowledge in environmental hormones and their negative effect on human beings, we decided to meet up with Professor Qingxiang Li, who is experienced and more familiar with this topic. After asking his opinion about environmental hormones, he suggests that the current environmental hormones are commonly existing in factory production and inevitably generated through the whole process automatically. Besides that, he also pointed out the environmental hormones cause the normal hormone regulation malfunction in the human body. In addition, they are very stable and difficult to decompose in the environment. So they can exist for a really long time, and it's hard for them to be removed. In addition, we also organized a meeting with another expert, Qiang Su, based on what she stated. However, there is no national examination for environmental hormones, and existing detecting methods nowadays can hardly catch up with the rapid development of manufacturing. The same problem exists with the testing of other types of environmental hormones. More of these commercially available estrogen tests require access to a laboratory and completion of an unaffordable and expensive set of procedures for civilians. Therefore, a more ideal test method is needed according to the professionals.
At the same time, we did a questionnaire on WeChat to collect more data. Please see the report from Lucia Yang below.
So, thank you, Brittany, and I will introduce the first questionnaire to you guys. So, in order to better understand views from public, we established the first questionnaire on WeChat to understand the public's views on environmental hormones as well, and through short text narratives to understand the public's concern about the impact of environmental hormones. In this questionnaire, we collected responses from more than 600 people from different industries, ages, and races in China and the United States. According to this chart, as many as 66% of respondents said that they have never heard of environmental hormones, which shows that most people do not know that environmental hormones may damage people's endocrine health. And after a short narrative, we asked everyone to choose the items that they most worry about might contain environmental hormones. Therefore, it can be seen from this icon that people have paid great attention to environmental hormones in daily products, especially food and personal hygiene products. The reason behind it is that the environmental hormone is closely related to daily life, and the damage to health is direct and obvious. In short, the need to test environmental hormones is clear and urgent. In other words, our research would have a great serve to the communities and have a great potential in the market if turned to actual products.
And our laboratory's project has also been carried out smoothly. Below, let's watch a video which introduces us to the methodology.
Here are our 5 components of ER sensor. Number 1, Lexi and terminal. Number 2, intent and terminal. Number 3, estrogen receptor. Number 4, intent C-terminal. Number 5, LEC C-terminal. We use overlap PCR to obtain a bunch of our full-length ER sensor DNA. We use PET28A to combine with the ER sensor DNA. Once these combinations of PET28A and ER sensor DNA are transformed into the E. coli cells, these E. coli cells will be able to resist kanamycins in the later experiments. These combinations were connected, then transformed into the E. coli DH5-alpha component cells. We place the DH5-alpha in the culture dishes that contain kanamycins. Thus, only the bacteria which contain the combinations will grow into monoclonal. We pick several monoclonals and cultivate them in the tubes of LV. Then, we extract the plasmids from these bacteria. We transform the plasmids into BL21 component cells and cultivate them in the tubes of LB. We purify the ER sensor protein, which is labeled with his tag. Ideally, these proteins will be the fundamental parts of the ER sensor.
At the same time, our teammates also designed a second questionnaire to future investigate to the public's purchasing intention.
And then I'll introduce the questionnaire 2 to you guys. So in order to better understand our market value, we have established a second questionnaire to test the willingness to purchase test kits and the expectations for such products. In this questionnaire, we collected more than 100 women of different ages and races from the United States and China. We divided these two groups of women into the following two sets: women aged 18 to 30 and women aged 31 to 50. So we got an icon that shows that women between 18 to 30 are more likely to spend 50 to 100, and most women between 31 and 50 are more likely to spend less than 50 on the test kits. Also, it can be clearly seen from the figure here that after being informed of the definition of environmental hormones and the potential problems these hormones bring to women, 48 of the 103 people interviewed were willing to choose level 3. On the contrary, only 31.1% of people are willing or very willing to use testing tools. Of the 160 women who were tested, 66 women were very willing or willing to purchase testing tools. Although there are still a large number of test subjects who are neutral on this issue, women aged 31 to 50 are still more concerned about buying testing kits than women aged 18 to 30.
At this time, our teammates have also completed the corresponding experimental work. Let's understand the result of the experiment and the next step of the experiment.
In this slide, these two pictures represent the results of our parts. The picture on the left shows the PCR results of LEC N-terminal, intent N-terminal, estrogen sensor, intent C-terminal, and LEC C-terminal. The picture on the right shows the plasmid extraction results from PET28A. After we obtain our full-length ER sensor DNA using overlapping PCR, we digested the DNA and PET28A plasmid using endonuclease, BAMH1, and Cy1. The productions were recycled and connected using T4 DNA ligase, then transferred into E. coli DH5-alpha component cell. In order to find out which monoclones contain the full-length DNA of the ER sensor, we picked 20 of them to verify using PCR. Only 2 of them contain the full length of DNA of the ER sensor. The picture in this slide shows the purification results of LEC. As is showed here, LEC was present in both sediment and supernatant. We also tested the reaction of LEC and Xgal. When different volume of X-gal were mixed with Lexi protein, different color presents. The doses of X-gal we used are 0 microliters, 2 microliters, 5 microliter, and 10 microliters. We then did an OMPG reaction testing using Lexi protein at different temperature. As we expected, the yellow color got deeper along with the temperature increased from 0 Celsius to 37 Celsius. This proved we purified and obtained active Lexi. We also express and purify the ER sensor protein, and SDS page electrophoresis was done to verify the results. This figure shows the results of protein electrophoresis respect to the ER sensor protein, which is labeled with his tag. The molecule weight of the target protein is assumed to be 176 kD, but in this figure, all the sample except the fourth one shows a molecule weight about 210 kD. Besides, the ER sensor protein expressed with or without the estrogen. We use the protein in our next experience. The ER sensor supernatants were used to functional test, and the result is shown here. The ER sensor can react with OMPG with estrogen was added or not. Although too little amount of active ER sensor, we can still make a conclusion. Our project and experiments work normally, and we can do some improvement in the future. Now, let me introduce the parts designed in our project. Firstly, we designed several basic parts, including promoters, tags, protein coding sequence, terminators. It is noteworthy that the ER sensor was composed by N-terminals of Lexi gene, N-terminal of VMA intane, human estrogen receptor coding sequence HER alpha, C-terminal of VMA intane, and C-terminals of Lexi gene. Then, we construct two composite parts, BBA K3525001 and 005, are used to express and purify ER sensor and Lexi protein in E. coli BL21, the E3, respectively, and test the function of these two proteins.
Later, we had an in-depth exchange with students from Shanghai International Middle School. Let's learn about it through Lucia Yang's introduction.
Thank you, Brittany. So I'll introduce the collaboration part. During our research, Enviro also had a deep collaboration with Shylock in the fridge, in parts developing. Since we were using the same plasmid, we exchanged the understanding of the operation on the plasmid. In the beginning, Enviro planning to use a certain method called homologous recombination, which is the method combine PAD28A and the target DNA unit by overlapping the same DNA box at the ends of both DNA sequences. With the specialized temperature and the special guidance materials, the ends part of both PAD28A and DNA units we created will open up the combine, forming a closed cycle. On the other hand, the SIF used another technique required a specialized enzyme, cut the end of the plasmid PAD28A, then combine with the developed DNA units. The way SIF develops their units provides us a brand new idea of how to connect our parts with the plasmid. While we encounter technical issues, we obtain their method and overcome the problems.
Through our communication and the above-mentioned experimental results, the students in our laboratory began to design a series of products.
Here is the step of using ER sensor. First, put the test sample into the beaker and add 10 ml hot water into the beaker. Then, wait for 10 minutes until the water is cooled down. Then dump the water into the ER sensor box. Afterward, wait for 60 minutes and compare the color in the box and the concentration standard card. At the end, dump the ethanol to eliminate all the bacteria in the box and deposit the box.
And now we'll introduce the social media part. To effectively build social awareness and enlarge our market, we released some articles through the internet on WeChat. We set up our official account and named it as our team name, Envir, to tell public about the negative impact of the environmental hormones and pay increasing attention to our product. The first article we published is about the background of our research, the brief introduction of our product, and the importance of our product. Furthermore, since we can hardly set up the offline meeting due to the situation, we presented several lectures online for high schools all over China and adults in the community. Our team wanted to inspire local people to make them aware of the importance of testing environmental hormones and how efficient our product will work.
Now I will introduce the safety consideration in the lab and about our product. As you can see, in the lab, following the instructions of your instructor all the time is one of the most important rules in the lab. What's more, you should always wear masks, gloves, lab coats, and a pair of safety glasses if possible. Remember to use the alcohol spirit to clean up your hands or gloves before you use the equipment in the lab. Oh yeah, unlike the labs in our schools, you should never bring your cell phones or headphones into the lab. And what's next is about our product. Remember, this will be important. First, do not damage the container and add other liquids into the indicator. Second, don't let your skin or organs get in touch with the indicator, which on the other hand means don't touch or drink or put these liquids into your ear and eyes. Third, you need to take great care of the waste. There are two kinds of indicators. For the equally one, you have to sterilize the indicator first in the case the equally won't harm the environment. For the normal protein one, you can just dump it with the container into the trash can. They are all safe to go. For the equally indicator, you can store it under normal room temperature, but for the protein indicator, you have to store it at 4 degrees Celsius. Thank you, Brittany. Now I'll introduce the next step of our experiment. The solution will show color blue depending on the concentration of estrogen in the environment. We will use technologies to determine the relationship between the concentration of estrogen and the degree of the blue after the reaction. And we will put a scale on the product to let people able to know how much estrogen are there surrounds them. During the research process, our team members have dedicated themselves into the project. You can find the attribution through these sites. Both dry team and wet team have done great jobs, and we won't complete our project without the effort from all of our team members. We also receive support from different channels. We would also like to say thank you to all the external supports.
Alright, our content is over here. Thanks to all students, instructors, and schools for their help to us. Thanks again to all judges, instructors, and students for your listening.